Hi, my name's Brianna, and I'll be talking about prenatal alcohol exposure and early alcohol experimentation in childhood. In Australia, 1 in 2 women consume alcohol prior to knowing they're pregnant, and 25% do so after knowing. For the majority of these women, they're consuming low levels, and as part of my PhD, I've been looking into the relationship between low level exposure and child outcomes. When examining 10,000 children aged 9 to 10, we found that any alcohol use during pregnancy is associated with increased odds of alcohol experimentation. And in a second study, we found differences in brain structure among exposed children. And this was the case even when we controlled for parenting factors, alcohol accessibility, histories of substance use disorders, school performance, and birth-related factors. So based on these findings, I wondered whether there was an underlying neurobiological role in early alcohol behaviors among exposed children. I decided to investigate the relationships between prenatal alcohol exposure, sipping alcohol in childhood, and brain volume and cortical area. I used baseline cross-sectional data from over 9,000 children in the ABCD study, and I controlled for a number of relevant covariates in the statistical models. In total, 1 in 4 children had been exposed to alcohol in utero, with the average amount of exposure being 27 drinks across the entire pregnancy. In line with my previous findings, children with any exposure were 66% more likely than unexposed children to have sipped alcohol by age 10. They also exhibited significantly greater total brain volume and greater total cortical area. Regardless of exposure, children with brain volume in the top quartile were 35% more likely to have sipped alcohol. And a similar association was observed for cortical area. So, is there a relationship between all of these variables? Do neural features partly explain associations between prenatal alcohol exposure and early alcohol use behaviors? I ran mediation models which showed that there was a significant indirect effect, meaning total brain volume partly explained the association. The same was observed for cortical area. In both cases, the proportion mediated was just 4%, which may indicate that other factors underlie and contribute to this relationship, such as genetic, epigenetic, and environmental influences. It's also possible that certain brain regions contribute more than others, and this should be further explored. So in conclusion, any alcohol use during pregnancy may play an important role in very early alcohol use experimentation, and long-lasting alcohol-associated changes in the fetal brain may be one of the contributing factors to these behaviors. We'll investigate this further when longitudinal data becomes available.